All right, so today I'm going to talk about X-ray crystallography. And as I was preparing this presentation, I I remembered just how deep the X-ray crystallography rabbit hole goes. Um, and I kept debating with myself what to leave in and what to take out and how deep to go into everything. And uh, I've I've prepared my presentation here, and I put quite a bit on here. I still don't know if I'm going to go into everything that I even have on this presentation, and so I'm going to try and work my way through what I have here. And if I don't touch on some things as much as you might be interested in, I have left quite a few um, different links in the description. Some of the things that I wasn't sure what to go into too deep were all the ideas of symmetry, of the symmetry and various symmetry operations, and determination of space groups. And I didn't know how much I wanted to go into how to solve the phase problem. If you've already watched my video on cryo-electron microscopy, which is mostly about single particle analysis, you will notice that these are all things that do not come up in that, which is one of the things that makes cryo-electron microscopy so much simpler, at least in my opinion anyway. And uh, probably why it's also increasingly becoming more desirable. There are a lot of issues with X-ray crystallography that don't come up really in cryo-electron microscopy. All right, so to get into it, up here I have an outline. Uh, so what we're trying to do with X-ray crystallography is find this protein model. And we want to do that by, uh, well, hitting a crystal with X-rays, so X-ray crystallography. So the first thing we want to do is purify our protein because we want to look at just a single type of protein. I'm not going to go too much into protein purification. That's a whole subject unto itself. There are multiple ways for protein purification, and it depends on the protein that you're trying to purify and its various chemical and physical properties. So let's just say that we have been able to purify our protein. Then the next step is we want to crystallize it, which would turn it into something like this. And to do that, we will use, well, a very common method is what's called the hanging drop method. Uh, hang, hanging drop method. And so we have our protein in a solution, and then we put a small drop of it onto a cover, and then place that cover onto a well, and it'll usually be in a multi-well plate like this. And so this is looking at a single well. And we have in the drop our solution with the protein. Then down here we have our solution absent the protein. And what happens is the solution up here is evaporating and and then condensing into the solution down here faster than this is going back into the drop. So this is slowly, you want it to very slowly uh, evaporate, and that will cause the proteins, because their solubility will reduce as their concentration increases, because you're losing solution up here. They will slowly start to form uh, into arrangements, into crystal lattices, and eventually these will turn into crystals that look something like this. There's all various different shapes that they come in, but this is an example of one of them. Then, uh, then what you do is you take your crystal, you scoop it out of one of these droplets. So you take this cover off, then you use this tiny little loop thing, and you want to get your crystal into that loop, and then you will place that in your your instrument, your X-ray crystallography instrument. So there are multiple different kinds of instruments. You can get sort of in-house ones that are that are not as good, but you can also go to what are called synchrotrons, uh, syn synchrotron, which is uh, a particle accelerator. So the one that I've used in my undergraduate research was the one uh, at Argonne National Laboratories, which is just outside of Chicago. And so what's happening is you're accelerating electrons around the particle accelerator. So they're going in that direction. And as the electron bends through this, it's releasing energy as X-rays. And so what you do is I think the X-ray would probably actually be sort of tangent to it. So you have areas around this where you actually collect the X-rays, and then you can use them, use that what's called synchrotron radiation to uh, to hit your crystal. And then what you get is a diffraction pattern that looks like this, or something like that. So that's what it shows here. Then once again, we have this phase problem, which once again, like I said, I don't know how deeply I want to get into, but there are methods for solving this phase problem. So as we'll see when I get into more detail about diffraction, uh, you have you're diffracting at different angles, and that will change the phases relative to each other, and you need to know these phases. And so we can use different methods. So there's uh, the multi-isomorphous replacement. There's the uh, multi-wavelength anomalous dispersion, and the molecular replacement, which is kind of the one you hope to have. It's a little bit easier. These other ones have a lot more steps, and there's a lot more. Well, I think it's mostly done with computers, but to understand it, there's a lot more sort of abstract mathematics and things like that. But anyway, once you have your diffraction pattern plus the phases, then you get your electron density, which will give you these maps of the electron density that you can model your molecules into. So you do real space alignments with that. You compare it with your diffraction data plus phases to do your refinement, and then you sort of iterate through real space refinements, and then these I guess Fourier space or reciprocal space refinements, and you kind of keep doing that uh, till you get through your validation. So you want your bond lengths and bond angles and all that kind of stuff to be in the uh, the allowed areas, such as you see in a Ramachandran plot. And later on, I'll go through more of the various validations. But once you go through that, then you get your final protein model. So we see here in this shorter version, we get our diffraction pattern. We go get our electron density map, and that's where you go through this process here, and then from that you can get your protein model. All right, and so if we want to, so we looked at this crystallization process. Now we want to think about the diffraction process, and for that, well, first I guess to look at the crystallization in more detail. When we're forming these crystals, our proteins, so this would be a single protein, comes together uh, into some unit cell, some cell that has various symmetry operations. So you can see a symmetry operation on this would be a rotation and a translation, or it might be an inversion. So an inversion, you sort of move through this twofold axis, and this part here inverts and ends up down here, and so forth. And so we have different symmetries that can be in this unit cell. And these unit cells form into this lattice with these repeating unit cells. Uh, and so these unit cells end up with what are called the Miller indices. And so what that would be something like we have a certain atom right here, and then if we go down to the next cell over here, we have that same atom here, and then that same atom here. And so we could draw a line like this, and then we have the same thing here, where we have that atom there and that atom there that are equivalent. And so we have another line like this, and this forms these various lines, because then you could have, say, the atom here plus the atom here plus the atom here, which gives us another line like that, like that. So we have these various planes here that give us our Miller indices. And so you can see here that there are various ways that we can arrange these planes like this, and we give them these number these number identifiers here based on uh, uh, based on where these planes are. So you see here what's shown in red is x is at 1, so 1 right there. But then all of the other ones uh, are at 0, and same here where y is at 1. The other ones are at 0, and z is at 1, and the other ones are at 0, and so forth. And so, like for this one, our y and our z are both at 1, and so on and so forth. Um, and so we can come up with these different number indicators for our planes in the crystal. And these planes in the crystal are what we are going to diffract off of. And so what happens when you diffract off of the plane is the closer together the plane is, the further apart the diffraction pattern is going to be. And that's why you can see here this, and that's sort of an intuitive way why they call it reciprocal space, because if our space in real space is separated by x, then in reciprocal space it'll be separated by 1 over x, where if this is getting larger, then this number here will be getting smaller. And so uh, as things get further apart in real space, they get closer together in reciprocal space. And uh, that kind of makes sense if you think about, just think about why, like if you have your hose on and you have water coming out of it, if the water's just coming out unimpeded, it's sort of just coming out in a nice stream like that. But if you put your finger on it and make the the opening of the hose smaller, so you put your finger on it and you're blocking some of it, then it's going to start spraying everywhere. And so that's kind of an intuitive way of thinking about when you reduce the size of these holes, then you're going to get much higher spread. It's going to become wider, whereas here we're getting this nice. If we just think about it right out of the hose, it's sort of a nice straight straight line with our larger opening. And so you can think about the spaces between right here, for instance, this space as an opening between it. Then you can see here there's this smaller space. And so that's going to cause uh, a diffraction that's more spread out, where here it's going to be closer together. Uh, and so the diffraction follows this Bragg's law. And so what that means is if if we have our planes here, then the incident X-ray beams are going to come in, and you see here that there is a certain uh, length that this has to be from here to here, so that once they're coming out, then we get constructive interference between the outgoing waves, and that is uh, so that we get constructive interference, which if we have waves that are in phase like this, then they will constructively interfere, give us a larger wave. If they're out of phase, then they will destructively interfere and we get nothing. And so as this angle here, which would be the same as this angle, as this changes, if it's going uh, this way or this way, then the path length difference between these two beams is going to change, and we're going to change the relative phases of them, and that will cause constructive or destructive interference. And so, uh, given these planes, there will be certain parts, if we're looking, if this is a screen that contains that's a really bad looking screen. So if we have a screen like this that's catching the different uh, outgoing beams, then you're going to have a part up here where the beams are constructively interfering, but as you go down, there's destructive interfering, then the constructive, and then it's going to keep going like this through the different angles. And down here we see if we're talking about different planes, then we're going to have uh, reflections off of these planes in different angles. There's going to be a different amount of difference between them in this triangle area right here that will allow it to have the same phase once they uh, reach this point right here where they have both become outgoing beams. And so so these are going to give us different reflections, and what we will end up with is a reflection pattern that looks like this. And so each of these little dark spots are reflections off of the different planes, the different Bragg planes, or the Miller indices, if you will. Uh, and sort of a look ahead at why we want to get this pattern is this through Fourier transforms will allow us to recapitulate the crystal or the structure. So the diffraction pattern is the Fourier transform of the crystal itself. And then from this we get these structure factors, which are the various little spots on here. Then through Fourier transform we can get our electron density from that. And so we want to think about what we need, what factors we need for our Fourier transform. And so the Fourier transform is shown right here. And what we need are these structure factors, and we need these positions here. And so these structure factors can be seen as the intensity of each of these dots. So the dots you can see have different, they're sort of darker and wider, or you might get these sort of lighter, smaller dots. And so what we want is this atomic structure factor, which is this part here of our Fourier transform. Um, so what we have up here is our structure factors, um, and so the structure factors we can obtain from the atomic structure factor plus the positions. So the atomic structure factor has to do with the way that the uh, the X-rays actually reflect off of the atom itself, and so this is the atom. And what we get here is, so this says that the charge distribution around the atom, which is just the electron cloud around the atom. So that's going to affect the size of the atom. And so that's going to give us different phase differences from the reflected X-rays off of it. And so we get our phase differences here, which is the difference between our incoming and outgoing uh, X-rays. Uh, so here we can think of, I put over here R, which we can think of as the location. So that would be the same as our our H, X, K, Y, L, Z up here. And so R is just the position of the atom. And so from that, we can get the individual contributions from the scattering fields. Um, and so with that, we can we can find our atomic structure factor here. You see this, the atomic structure factor, and that is one of the factors that we need in determining our structure factors for the full crystal. And so that is seen in the intensity of these spots, and then we also want the position of them. The intensity of them, you can see here, is dependent on how the X-ray is diffracted off of the atom, uh, like you can see here, and so, or it depends on the angle in which it's diffracted off the atom. So you can see here with the angle down here, our structure factor is dependent on the number of electrons in the atom. So if we have something like cesium, which has 54 electrons, then we're going to get this level we're going to have this atomic structure factor uh, when we're coming right off of the atom like that. But as we change our angle, then the atomic structure factor is going to be reduced until we get back here uh, where it's 180-degree diffraction off of the atom. And so with this uh, with this intensity here and the position in our diffraction data, then we can start determining these structure factors. And the structure factors start telling us things about our crystal. And so the first thing we want to do is determine the crystal system and the unit cell dimensions. And so our unit cell dimensions are based on the lengths of the unit cell. and the angles of the different components of our unit cell. So the unit cell, as we talked about up here, is one of these. So one of these is a unit cell. You can see that down here. And so the unit cells in a crystal lattice are going to have different dimensions. And the dimensions of it we can determine from the diffraction data, uh, and we can get what are called the Lowy groups. I don't know if that's how you pronounce it. I've only ever seen it written. And so we get these Lowy groups, which tell us different things about the lengths and the angles in our unit cells, and they have these different names for the Lowy groups. Um, and so to obtain the Lowy groups, we actually want to look at the symmetry of our diffraction data, and the symmetry. So you can see up here, this shows a rotational symmetry. If you rotated this 180 degrees, you would end up with the same diffraction pattern. Um, and so we can obtain the the Lowy groups from the symmetry of our diffraction patterns, and we can determine whether it is a central symmetric structure or a non-central symmetric structure. Uh, and so this is one of those places where I debated just how far to go down the rabbit hole of symmetry. You can There are full textbooks on these ideas of symmetry and point groups and things like that. So I don't want to get too bogged down into it. Um, so I think so. I think I'm just going to go to this table that I made here. And so what we're looking for are the space groups, which I have the list of those right here. So we can see we get the Lowy groups, or Lowy classes rather. And from that, we can obtain the space groups. So we want to get the Lowy classes, which have the rotation and inversion symmetries. And rotation and inversion symmetries are the ones that are centrosymmetric, because there is at least one spot that's not being changed. So if we had something like this, then we know that that's rotational symmetry. We can rotate this 180 degrees and it gives us the same thing. It's inversion symmetric because you can invert this over to here, this over to here, and we get the same thing, but this is not moving, so it's centrosymmetric. Uh, but then we also want to know, uh, so if we have something like this, uh, it would also be in a crystal where there is something else in the crystal next to it like that. So this would be a unit cell, but then we could translate that unit cell down here and it would completely overlap with this. And so that's how we can get what are called our Bravais lattices. Once again, Bravais. I've only ever seen that written. So Bravais lattices, uh, and these are obtained from our diffraction pattern symmetry, like I was talking about down here, and their diffraction pattern positions, uh, and from the intensities of them. Then to get our space groups, which is talking about both or the rotation, inversion, and translation, plus what are called screw axes and glide planes, which are another type of symmetry. So that's how we get our space groups. Um, like I said, I've left some. links in the description that go into more detail about this idea of symmetry, but I just I don't I don't want to get too bogged down into it. It kind of goes off in the weeds. It's a very in-depth uh, it's a very in-depth sort of subject matter, and there's just a lot to it. And so uh, I don't want to I don't want to get too bogged down into it. Uh, but so once we have our space groups, then we know something uh, about the symmetry of our crystal structure, the crystal lattice. Um, and then from there, we can use that to determine the position of atoms in the crystal. To do that, we need to think about the phase problem, and so uh, the intensity of a peak in our diffraction pattern tells us. So once again, let's go back to this Bragg diffraction. So the peaks here tell us something about the planes here, but we see that uh, even though these are in phase, this has gone one extra phase or one extra wavelength. And so what that does is uh, it obscures the actual phase information of our structure factors. And the phase can be thought of if we're uh, so the phase, the structure factor is determined uh, by a complex number which looks like this, and the structure factor is is so the intensity is the structure factor squared. And so when you square a complex number, you are multiplying it by its complex conjugate, which in this case would be this. And so what you end up with is a squared plus b squared. And so this this phase difference is lost when we use our intensities to find the structure factors, and the phase is so you can see here a phase shift. So the phase shift can be thought of in a complex space as uh, the angle that the vector, this vector here is pointing, but we don't know the phase, so this could be anywhere along the circle. This phase shift could be offset by uh, any number of degrees. So the phase shift between A and B here is shown as an angle here between A and B. Uh, and so we see over here that our structure factor, so if we have a reflection off of this plane here, and then we have another plane of atoms that are have this vector away from our plane here, so they have a plane like this, we're going to have reflections off of this plane as well, and that's going to create a phase difference between the two planes. And so we will only see this structure factor here. So we will see this this intensity here, but we don't know what these different phases are from the two different planes that we were reflecting on up here. And you can have multiple different planes, this shows, with all their different phase angles. So we will only know what this F at HKL is right here, but we won't know all of the different phase differences here. Um, and so what we want to do to be able to find those phases is we so what we're talking about here. I mentioned earlier these three different ways of obtaining the phase data. So we're talking about here multiple isomorphous replacement. So what we do is we get the diffraction data from our crystals, uh, either with and without some kind of heavy atom. In this case, it's talking about platinum bound into our crystal. So it's isomorphous because these heavy atoms in here are not changing the uh, the structure of our protein, and so we can find the position of our heavy atoms by um, by subtracting the diffraction data from our so from so the FPH is the structure factors from our proteins um, with or without the heavy atoms, and the FP is with the heavy atoms. I think that's the direction it goes. So let's look over here. So the all right, so the FP is with is without the heavy atom. So it's heavy atom minus without the heavy atom. And so what you end up with here is, so I mentioned up here, if we don't know the phase, then it can lie anywhere in this circle. Our phases can be offset by any degree, by any amount of phase offset up here. So our angle could be anything. And so that's what this shows here is if we're just looking at the FP, which is our crystal structure without the heavy atoms, then it can be in this circle here. And then if we get the structure factors with the heavy atoms, then we actually uh, we get yeah. So the FH is determined by the real space positions of the heavy atoms. Yeah, so this is without the heavy atoms. Then with the heavy atoms is the circle made. So this is the origin right here. And then we just make a circle like that with the heavy atoms. Um, yeah, so I think pH is protein plus heavy atoms and then protein. So uh, so then we end up getting these two circles like this, which are the various phases that you can get from the the heavy atoms, and from that we can find out where they overlap here, and you see that we end up with two places where we overlap. So we have two different possible phases that we have, which are these two alpha p's, and from there this is what's called single isomorphous replacement. So we use the heavy atoms in a single place in our proteins. Then we would want to go to multiple places. And so we have the two different heavy atom crystals. So that this was circle here was from our first one, and this other green circle here is from our second one. And then where they overlap here is now we know what our phase is. It's this alpha right here. So we could determine the phase that way. Um, I had over here, so I was, this is one of those things I was debating about going through was this idea of the Patterson function and the Patterson maps, and the Harper planes. And once again, this just gets into all sorts of, this just goes down a huge rabbit hole of different abstract mathematical ideas of taking your Fourier transform without the phase data, and then you can use the different Patterson different maps to get the vectors, and that will give you the placement of your heavy atoms in the crystal. I think I'm going to skip over that stuff. Uh, Once again, I have links in the description if you want to look into that. It can be kind of long and confusing and probably not all that enlightening for our purposes right now. Uh, so, long story short, what we end up with is uh, is we can determine the electron density from the information that we get from our diffraction pattern. Once we know the space group, which, like I said, was determined by the symmetry of this diffraction pattern, the symmetry of the intensities. Uh, so that's what you can determine just from the diffraction pattern, but then we also need to solve for the phase problem. And from that, like I said, long story short, we can obtain the electron density, which you can see here. Um, so this is actually a really good electron density. You can actually see this cyclic uh, side chain, which would be a phenylalanine. You can see little bulges where the carbon atoms would be. You can see the little hole in the cyclic part of this, the benzene group on this phenylalanine. You can kind of see here where the side chain would go down and connect to the uh, the main chain of the protein. Um, and so with the electron density, so we can see that here in this mesh, you can model in your atoms into the electron density and form the side chains of your amino acids in the protein. And so here we see in this purple is the well modeled uh, electron density. What we want to pay attention to here is that we have this green mesh here and this red mesh here. And what that means is that the red mesh is, you can see here we've modeled this atom down here from our side chain to be pointing in this direction. And what this is saying is that we've modeled our atom here. So we're saying that we think there's electron density here, but this red is telling us our diffraction data does not show that there's electron density here, but it does show there's electron density here. And so what we would want to do is do a real space refinement and actually change the orientation of this atom here, and then we could uh, we could compare that to our reciprocal space through a refinement, and what that would do is end up telling us that so then we would probably, this right here would end up being purple and this would go away. Or what you see here might be the case where you can actually have what are called different rotamers, and so we could model this in two different ways. And so half of the electron density is modeled in from this right here, and half is modeled from that right there. And so that's why uh, we might end up having this red here, because it's saying, what the program is saying is, I'm not detecting as much electron density here as what you were trying to model in. And so we could then change it to a model like this, where uh, maybe it has two different rotamers that it could possibly be in. Um, and so from here, once we have the electron density, we want to start doing our modeling, which, as I talked about before, was that sort of circular path of doing real space refinements and then comparing that to our data. And so that's what it's showing right here is we calculated our 3D electron density map, which was the Fourier transform of our diffraction data with the phase information. Then we build in our atomic models into it. Then we want to refine that against the experimental data. Then uh, this shows it going over here, but what you would actually do is go here and then do real space alignments, trying to fit your your atoms into the electron density. And then once again, once you fit that in there, and what you're doing in the real space alignments is you're trying to fit it into the electron density while also maintaining the bond angles and bond lengths and allowed rotamers and and things like that. So you want to fit it in here as well as you can while not having your angles and lengths and everything outside of the allowed values. And then from there you do your alignment against the experimental data. Then that will tell you whether you are uh, whether you are trying to model too much electron density into different parts. So then you have to do real space alignments, and there's this sort of circular process which it can it can take a while. Um, and then so what you're looking for when you're doing these refinements as well is you're looking for whether your observed and calculated data, how well those are fitting together, and that's what's called this R factor. So the R factor is telling you how well your model is fitting the data. And up here we want our, uh, our R factor to be less than zero point 0.2. So you can see here, this is from a crystal structure that I obtained in my undergraduate research. You can see we got an R value here of 0.180. The R free is using some data that was sort of especially set aside during this refinement, so that then you can use that data that was set aside to compare to your data, uh, so that it's not sort of just being circular of comparing refined data to refined data. And so that's always going to be a little bit higher, um, because it's more stringent. Um, but once again, you want that to be in the range of zero point 0.2. Um, and so that's calculated from our observed structure factors minus our calculated structure factors over observed structure factors. Uh, and so right here are the different ways that that we validate our data. And once again, this is from the X-ray crystal structure I got of a protein which was called OXA 239. It's a clinical variant of an enzyme that bacteria use to break down uh, antibiotics to confer on those bacteria antibiotic resistance. Uh, and so we see we got our specific space group here, which we determined from the data. We got our cell constants, which was the uh, the Lowy group. We got our resolution. So resolution is how well you can tell the difference between two different atoms. And so say you have two atoms here that are bound together. If you have high resolution, then you'll be able to see the electron density differences between the two atoms, which was what we could see here, where you can see there's bulges where the atoms are. If you have worse resolution than if you have two atoms bound together, you're going to get something that's not able to really determine the where one atom ends and the other one begins from the electron density. And so this resolution, which is in angstroms, is telling you uh, the resolution at which you can tell the difference between two things. So if this has a bond length here of, say, uh, let's just call it 2 angstroms, and you can tell the difference between them, then you have a resolution that's lower than 2 angstroms because you can tell the difference between two things that are less than 2 angstroms apart, whereas here uh, it would be higher, the resolution would be larger than 2 angstroms because you can't tell the difference between these two atoms that are 2 angstroms apart from each other. And so my data was 1.81 angstroms, and I will show my data in a program here in a little bit, and you'll see that my data is not as good as this. This right here would be extremely high resolution data. This would probably be, uh, probably less than 1 angstrom. Uh, Yeah, and so you can see here I wrote a description over here for the R merge and R sim. They're metrics that give an indication of the agreement between intensity measurements, so intensities from our diffraction data. Here is our signal to noise ratio. This just tells us the program I used for this refinement here against the experimental data, which is a program called RefMac, and that's sort of a black box program. You just kind of put your data from the real space alignments in, and then it just sort of spits something back out. These are the R factor and the R free again. Uh, and then so we have the R free test, which just tells us this number of reflections. So this B factor, which you see here, so that's the Wilson B factor, but then we also have the average B factor here. And the B factor, so I would think of that as sort of a floppiness factor. So I actually have stuff up here about the B factor. So you see the B factor, the higher it is, the more sort of uncertain you are of your uh, backbone being in a position, say, well, this is one that is easy to pick out. So this has a high B factor, which means that this is our protein chain here. So this means that the protein chain is sort of flopping around here. So high B factor is like I said, it's sort of a floppiness factor. And you can see that even though we have down here this sort of average B factor, which was 41 for my structure, um, what you actually see is the B factor changes throughout the protein chain, and that's we can see over here. So we can see the B factor by residue number. These are these images were taken from different things, so these are not the B factors that we see here. I just wanted to illustrate that you can get B factors for the different residues here on your protein chain. So uh, this is the C terminal domain over here. This is the N terminal domain rather. This is the C terminal domain. So these are the amino acids in our protein, and you see that they can have these different B factors. And so that's what the B factor is this, like I said, this sort of floppiness factor. And the B factor changes the atomic structure factor uh, from our. So before we had our FHKL, and we determined that from the structure factor, the atomic structure factor, which was our F, then our E of the positions, which I'll just write as R rather than writing it out. But we have to use our B factor to give a correction to our atomic structure factors because the atoms, like I said, are flopping around, and so we can't just say that our atomic structure factor is based off of this sort of nice rigid sphere because the sphere is sort of moving in all different directions, and so we correct that with this B up here being our B factor, and so we actually will use this here, which is our atomic structure factor, corrected with the B factor, and that's sort of the importance of the B factor. We have our F observed and F calculated correlation. Uh, so that's 0.96, so perfect would be 1, so this is pretty well correlated. L test for twinning. So twinning is just something that happens if you have sort of two. So if you have a crystal like this, and then you have another crystal that's sort of attached to it. So you get these twin crystals. So if X-rays are coming down onto it, then it's going to diffract different off of this crystal than it will off of this crystal. We see that the L test for twinning. So estimated twinning, no twinning to report. L test for twinning. So the theoretical values for untwin is 0.5 and 0.375. You see mine is 0.49, 0.32, so that's pretty close. So it doesn't look like there was any twinning in my crystal. The bulk solvent, which I have written here, B sol and K sol are parameters in the formulation or estimation of bulk solvent contributions to the structure factors. So what that means is we have our crystal, and so say this is our unit cell on our crystal, and we have our protein in here, and well, it'd probably be another protein, so maybe there's multiple proteins in our unit cell, but there's also going to be water molecules all throughout here. So the water molecules are going to contribute to the diffraction data because water molecules are also present in our unit cell. And so this is just a measure of the contribution of our water molecules in the diffraction data. So these are all the various things that we can use for validation of our crystal structure. I show up here as well that we have, so this is our R-free, and the dark section here is telling us the percentile relative to all the X-ray structures, and this empty one here is telling us the percentile relative to X-ray structures of similar resolution. So resolution in in the 1.81 angstrom range. And so you can see that mine, the R free is in the the better area rather than worse. Clash score, which is just telling us if we have two side chains from different parts of the protein. Um, Clash score just means that they're a little bit closer together than what we would normally like. Ramachandran outliers are the bond angles. So you see on mine there are 0 Ramachandran outliers, which means that as I went through the real space alignments, I was able to get the bond angles, the phi and psi bond angles. That should be phi. Wow, I can't draw phi and psi angles. If you remember that from the the protein structure video that I made. So Ramachandran outliers, there are no outliers. Uh, these side chain outliers, which would be like the rotamers that I was talking about. So there are allowed rotamers, and then uh, real the real space are it's the real space R values. So these R values are determined uh, in Fourier space or reciprocal space, and these R values are determined in real space. And so you can see these percentile rankings for my protein, which I will actually take a look at in a protein or in a program called Coot here in a minute. But first, let's do a quick little recap of what we went through here. So I talked about we to obtain a crystal structure, we first purify our protein. There's various different ways you can purify protein. There's crystallization, and I did not go in depth in crystallization either. So I sort of just talked about this hanging drop method. There are other methods, but uh, the depth that we could have gotten into are looking for the various solutions that you can use to actually form crystals. So you adjust the pH and the various salt concentrations and everything of your solution, and you want to test. Sometimes you can test thousands, tens of thousands of different solution conditions before you find the right one to form crystals. You could probably do an entire uh, 7-year PhD program just trying to find how to form crystals of a certain protein. So that's definitely a limitation of X-ray crystallography that you don't find in cryo-electron microscopy. But so crystallization, then we went and found our we we hit it with X-rays to get our diffraction pattern, which we then use for the data processing. We needed to solve the phase problem, which we could then get our electron density map, and then we could do our refinements to uh, with our refinements, we want to fit all of our atoms into the electron density map, but we also want to make sure our bond angles and so forth our validation is good, and then we can get our final model. And crystallization was aligning the the proteins into these unit cells, which have a certain symmetry. You can see here that we might have something like a rotational symmetry or an inversion symmetry. And then these unit cells align into a lattice here, which we can then get our various planes from, which give us our Miller indices. Uh, we get these reflections off of the different planes, which are what give us the diffraction pattern. From the diffraction pattern, we get our structure factors, with which the structure factors are based on this atomic structure factor plus the position of the structure factor. And the atomic structure factor had to do with the electron density around the atom. We determined the space group of our the space group of our crystal structure, which gives us the various symmetry operations. We went and we found the phase information, which we did by getting the crystal structure of the protein with and without heavy atoms in it, and with and without the heavy atoms, the FPH minus FP gave us this FH, which is what we got here, and that we could use as a new origin with which to make this circle of the different possible phases. And then we can get the overlaps, which gives us the phases of our protein. And then doing the multiple isomorphous replacement, we can then narrow it down to a single possible phase for our crystal structure. Like I said, I did not go into the Patterson maps or anything like that. That's a whole other topic itself. And so we can use this phase data plus our space groups to determine the position of atoms in our crystal. And from that, we can actually get the electron density map. And from that, from the electron density map, then we could model in our atoms. And from there, we can generate our model. And I will show you what that looks like here in just a second. Okay, so this is a crystal structure that I obtained from in my undergrad research, and what you can see here, the yellow is the protein structure. As I move it around, you can see that there are actually two proteins here, both facing in different directions. That is because this is from the unit cell. And so these are symmetry related to each other. So you can tell just from sort of, if you look at this phenylalanine here is the same as this phenylalanine here. So there is if we reflected this over, and then you can see there's somewhat of a twist. So if we reflect it over and then twist, then we would get it to here. So that is the symmetry operation that we would perform on one of these in order to get the other one. But what we want to look at here is, you can see in the blue mesh the electron density that I obtained. And as we were just talking about, the electron density is obtained from the diffraction data. And so what you can see here is, in the yellow and blue and red, I've modeled in the atoms. And what you can see is, for instance, right here we have a leucine. So right here is the alpha carbon. This is the beta carbon, and then we go up here to the branch chain. And you can see in blue we have nitrogen, and in red we have oxygen. And so right here we have our peptide bond. In the N-terminal direction. Here we have our peptide bond in the C-terminal direction, and you can move to the, so you can move one one at a time, one amino acid at a time down the structure here. So here we have an isoleucine, then we can move down and we have a histidine uh, and a glycine, or no, this is a glutamine rather. So you can see up here we have the nitrogen and oxygen on our side chain. And so what we do when we're in here is we can do what are called real space alignments. So we will real space align from here, and then this tells us if our real space alignment has it's changed our bond lengths and the bond angles, the planes and chirals, and you can see in the sort of more ghostly white where the new one would be, and you can sort of move it around and try and fit it into other electron data. And as you do, this will tell you you're changing in the bond angles, and when we have yellow, that means it's not very favorable. And so we can, I'm trying to get it to sort of go up into that green there, but doesn't seem to want to. See here, it's kind of gone off into nowhere. And so that's how you do real space alignments. So what you would do if you were trying to fix this is you'd want to model in all these atoms and then try to align them into the electron density. Let's see, there I was able to shift that over there. You could try shifting it over there. And then what'll happen is if you accept that and then you run it through your refinement that compares it to the diffraction data, then it will tell you whether using either green or red, whether it's the diffraction data, the sort of the observed data is it corresponds well with what you're trying to model in. So by modeling in an atom right there, say, I'm saying I'm calculating that there is going to be this electron data right here, and then if there's observed electron data there, then it will turn blue and say, yeah, that corresponds. You said that there was going to be electron data there, and that is what was found. And so then it turns blue like that. You can see there's green here where there's possible electron data that we are not actually putting in, that we have not modeled in. We can try and move this over into the green there. Doesn't seem to want to. It's probably too far outside of the bond angle range, and hence why it was not modeled there in the first place. I mean, you're going to end up getting some noise. So green means that there is electron density data that was found in the actual diffraction data. Red means that there's less electron data than what you are trying to model into it. But this is likely just from noise. So we talked about how the water in the crystal also contributes to the diffraction data, and that's what these little red doohickeys here are. So they look like this because the electron data you're seeing is almost entirely from the oxygen, And so they just kind of make this cross to show that the hydrogens could be pointing in all these different directions because hydrogens don't show up real well in the electron data, which you can see just from here. So we know that something like this carbon would have two hydrogens on it, but you don't see any electron data from the hydrogens. Uh, so yeah, this is the electron data compared to what we've modeled in. So uh, you just want to try and align this, and then you end up with so then you end up getting your refinement models, and then you get your validation here. And this is all stuff that you would find on a website called the Protein Data Bank, PDB. Uh, so it's a database where you upload your structures onto the web so that uh, then they are available to other scientists. And all of this validation parameters here are things that you would find on the protein database. And yeah, so that's X-ray crystallography for you. Um, just to kind of go through that one more quick time, we get our crystal, we obtain our diffraction data from that, from the positions, intensities, and the phase data that we have to account for. We can get the electron density map, we model our atoms in, and that gives us our protein model. Uh, I I apologize if some of this was confusing because like I said, I glossed over a lot when it comes to the symmetry and the space groups and the phase problem. But you could fill an entire textbook, an entire 2 semester college course just talking about this stuff. And uh, I mean, this playlist I'm making is, you know, at least nominally talking about nutrition. So going off into the abstract mathematical weeds on this just is not going to be all that useful. Um, I wanted to talk about this. I mean, both cryo-EM and X-ray crystallography are things that I have done in my own research, and so they're kind of near and dear to my heart. And I'm the one giving these lectures, and so you have to put up with the way that I do it if you are watching these. But also, I think it's just interesting because in a lot of biochemistry textbooks and stuff, you will look at these structures. They show you this part here, and they can tell you all the different things about it. But it might give you an idea, this false idea in your head that to see what this looks like, you just sort of put it on a microscope slide and just zoom in really far, and then you'll start to see these things. But that's not how it's done. X-ray crystallography is sort of the old school, the OG way of doing it. Cryo-EM is starting to become much more popular, and it's becoming a lot more desirable. Like I said, because you don't have to deal with a lot of this stuff, particularly the phase problem, which if you don't have structures already available for doing molecular replacement, which is sort of the easy way of doing it, and to do molecular replacement, you have to have uh, phase data from structures already obtained that are similar structures to the one you are working on. So maybe if you're working on just uh, a mutation, which is actually what mine is, my structure here is just it has 3 point mutations, 3 amino acids different from a structure that was already obtained. So I was able to use this molecular replacement to get the phase data. If you don't have that, then you have to go through all the rigmarole of doing these heavy, you got to get crystals with the heavy atoms, uh, ideally in multiple positions, so you can get this multiple isomorphous replacement. You have to get it without the heavy atoms, so you have to get multiple crystals. Then you have to get good data from the multiple crystals. Uh, there are ways to do it without multiple crystals. The MAD, the multi-wavelength anomalous dispersion, which is just uh, doing this with wavelengths of X-rays that are in the range of the resonance of the heavy atoms. I'm not going to go too much into that, but uh, the finding the phase finding the phases can be a whole exercise in itself. Like I said, I didn't even go into these Patterson functions and Patterson maps and things like that that goes even further off into the weeds. Uh, and so X-ray crystallography it uh, can be quite laborious. Like I said, there is the crystallization process, which can be very laborious, and uh, there's uh, different kinds of proteins that are extremely hard to do, like membrane-bound proteins and things like that. Then finding the phases is another huge issue, which makes the cryo-EM look a lot more desirable. If you watch my cryo-EM video, you'll see that it's a lot simpler. It has its own ups and downs, uh, which which I got into in that video, um, but yeah, X-ray crystallography is sort of a tried and true method, and you can get really good structures with it. Cryo-EM is still sort of in it's still an emerging technique, particularly the single particle analysis, and so it's not as well developed. This process here is sort of a tried and true, well developed sort of process, and you can get really high quality structures from it where cryo-EM is only just starting to get structures that are on par with what you can get from X-ray crystallography. And anyway, I've been talking long enough and even repeating myself, and so uh, I will I will call this video good enough, and hopefully you found it at least somewhat enlightening, even if you're confused about some of the various weeds that we could get drawn off into. But I will see you in another video.